Merhaba, herkese iyi akşamlar. Ben doçent doktor Ayça Sayı Yazgan. Türk İmmünoloji Derneği olarak düzenlediğimiz İmmünoloji Aydınlatıyor webinar serimizin 29.suna hepiniz hoş geldiniz. Bugünkü konuğumuz Amerika'dan bizlere katılan Doktor Mustafa Akkoyunlu. Konuşması öncesinde kendisini sizlere kısaca tanıtmak istiyorum. Doktor Akkoyunlu, Ankara Üniversitesi Tıp Fakültesinden mezun olduktan sonra doktorasını İsveç'te, Lund Üniversitesi'nde Doktor Arne Forsgren'in danışmanlığında tamamlamış. Ardından doktora sonrası çalışması için Amerika'ya giderek Yale Üniversitesi'nde Doktor Errol Pickerick'in laboratuvarına katılmış ve çalışmalarına burada devam etmiş. 2002 yılında Doktor Akkoyunlu kendi laboratuvarında FDA yani Amerika Gıda ve İlaç İdaresi'nde Center for Biologics Evaluation and Research merkezinde kurmuş. Halen kendisi çalışmalarına laboratuvarı burada devam etmektedir. Doktor Akkoyunlu laboratuvarında beyin empati aktivasyonu faktörü BAF'ın bağlandığı reseptör olan Taki'nin hücreye özgü fonksiyonları üzerinde çalışmaları yürütmektedir. Kendisine verilen birçok ödül var. Bu ödüllerin bir tanesi de 2016 yılında aldığı Siebert Bilimsel Başarı Ödülü'dür. Doktor Akkoyunlu bugün bizlerle Taki'nin hastalık immunopatogenezindeki rolleriyle ilgili çalışmalarını paylaşacak. Sorularınızı bizlerle YouTube üzerinden paylaşabileceğini sizlere hatırlatmak istiyorum. Paylaşabilirsiniz sorularınız varsa. Türk İmmünoloji Derneği Yönetim Kurulu adına Doktor Mustafa Akkoyunlu'ya davetimizi kabul ettiği için tekrar ben çok teşekkür etmek istiyorum. Ve sözü şimdi çok uzatmadan kendisine bırakmak istiyorum. Mustafa Hocam buyurun.
Evet, çok teşekkür ediyorum Ayça Hanım. Türk İmmünoloji Derneği'ne de çok teşekkür ediyorum. Davet için ve konuşmama başlayacağım. Konuşmam İngilizce olacak. Share screen görülüyordur umarım. Okay, so today I'm going to talk about how Taki role has evolved in time, and as Ayça has mentioned, my lab focuses on the role of Taki in different disease settings. First, I would like to introduce where I work. I am a member of Laboratory of Bacterial Polysarchites under Center for Biologics Evaluation and Research. In LBP, we focus on vaccines composed of polysaccharides, and these vaccines are intended to prevent diseases caused by some of the bugs that are listed here: Neisseria meningitis, Streptococcus pneumonia, Group B Streptococcus, and these diseases are bacteria are causing diseases especially in newborns, debilitating diseases such as bacteremia, meningitis are important outcomes of these diseases. And one common property of these bacteria is that they're covered with a capsule, and this capsule is composed of polysaccharide moieties. You can see examples of two bacteria here, Neisseria meningitis and Streptococcus pneumonia. Even though they're gram-negative and gram-positive, they both have this common feature, capsular polysaccharide. The capsular polysaccharides have several virulence properties, so they protect the bacteria from host. For example, they can inhibit complement activation, C3b, they bind to C3b. They can inhibit phagocytosis, and they can, I'm sorry, I'm pausing because there are things popping up here, like start my video, but sorry about that. Anyway, so another property of these capsular polysaccharides is that their expression levels can change. And as the expression levels increase, they become more invasive. And in addition to these, our laboratory has contributed to this field by showing that polysaccharides from Meningococcus type C can bind to CD14, which is a co-ligand for LPS, and they can shield from bacteria, can shield from host recognition. And this study we conducted with Can Kocabaş, who was a fellow in my lab at that time and who is a professor of immunology and allergy at the Muğla University now. And in addition to being virulence factor for the bacteria, polysaccharides are also the Achilles heel for the bacteria. Why? Because antibodies directed against polysaccharides are very effective in killing the bacteria. And this was recognized already in 1940s. You can see from this publication that immunization could elicit protection against pneumococcus. However, there are important shortcomings of polysaccharide vaccines. First, they are T-cell independent type 2 antigens, which means that T cells, unlike proteins, don't help to the immune response to polysaccharides. What this means is that in adults, when you immunize, you get short-lived antibody responses, IgM and IgG3 especially, and there is no memory response to booster immunization. An example of this vaccine is Pneumovax 23, which is a pneumococcal 23-valent vaccine used in aged population. In contrast to adults, infants don't respond to these vaccines at all. So it's an important shortcoming, especially when you think that the bacteria such as meningococcus and pneumonia target the early age population. So how does an antibody response to polysaccharides start and mount? And you can see here is a representative B cell and then B cell receptor. So polysaccharides are multivalent antigens, repetitive units of the same polysaccharide structure. They engage the BCR, but this engagement is not sufficient to activate the B cells. There needs to be a second signal. The need for second signal has been recognized for a long time, but this molecule was obscure until about 20 years ago. Two important papers published by generating Taki knockout mouse showed that this second signal was Taki. Taki has two ligands, BAF and APRIL. So in addition to BCR engagement, BAF and APRIL provide the second signal, and now the B cells can produce antibodies and even class switch. So what is Taki? Taki is a member of BAF and APRIL system. You can see in this cartoon that BAF and APRIL primarily produced by myeloid cells, and they can be cleaved by furin convertase enzymes. They can be trimers or sextamers for BAF. APRIL can also form multimers, but this formation of multimers happens as a result of engaging the heparan sulfate proteoglycans. So there are three receptors. BAF receptor is very important, and BAF receptor only engages BAF. And this is important because it provides B cell survival signal and IgG class switch. Taki, which is transmembrane activator, calcium modulator, and cyclophilin ligand interactor, is important for, as I said, for T-independent type 2 immune responses, and also provides IgA and IgG class switch. We know this from Taki knockout mice. They do not respond to polysaccharide vaccines at all. And also, more recently, combined variable immune deficiency patients with Taki mutations have been identified, just like the Taki knockout mice. These patients don't respond to polysaccharides very well, and they can suffer from recurrent Haemophilus influenza and pneumococcus diseases. BCMA is the third receptor, which is more obscure, only expressed on plasma cells. And we became interested in the Taki because, as I said, the knockout mouse don't respond to polysaccharides. And then given that newborns also don't respond to polysaccharides, we asked the question, what is the status of Taki in newborns? And to answer this question, we first measured the Taki levels on B cells. Here you see a flow cytometry with the gating of B220 and Taki. In adult cells, there's abundance of Taki expression. We found that newborns express very low levels of Taki. So what this means is that when BCR engages the polysaccharide in newborns, but because the Taki levels are low, a lot of BAF and APRIL cannot bind, and as a result, this reduced expression of Taki may be responsible for the unresponsiveness of newborns to polysaccharide vaccines, and we published this more than 10 years ago. So if the Taki levels are low, can we then increase Taki levels in newborns and render them responsive to polysaccharide vaccines? And we can test this because previously we have shown that adjuvants such as CpG can increase the expression of Taki and render these B cells more susceptible to BAFF and APRIL. You can see here that the Taki levels can double in adult mice B cells. And we did the same thing with newborn, and indeed in newborns also we can increase Taki levels. They come almost the same levels as the adult. And this means that now we can see the increased Taki expression, and then response to BAFF and APRIL is also increased. Indeed, we tested this, and you can see that polysaccharides given with PBS don't induce any antibodies in newborn mice, but when we immunize newborn mice with CpG containing vaccine, polysaccharide vaccine, we can now mount antibody responses. We were also interested in the question that why polysaccharides in general are weakly immunogenic antigens. We know that they're T independent, but is there something else here? So we incubated B cells from adult mouse with meningococcal type C polysaccharide, and then measured the expression levels of Taki. Here you can see the control, and on this side, when we incubated B cells with 2.5 microgram per ml, we already started to see a decrease in Taki expression. And as we increased the polysaccharide concentration, the decrease in Taki expression was more pronounced. What this then meant is that after encounter with the polysaccharide, the B cells appear to downregulate their Taki expression, and this then results with a diminished response to polysaccharides. So these two studies were our contribution to the role of Taki in B cells. We showed that newborns express low levels of Taki, and this seems to be the reason why they don't respond to polysaccharides. And then also we showed that meningococcal type C and group B strep type 5 polysaccharides can actually downregulate the Taki expression and evade this seems to be a virulence property for certain encapsulated bacteria. And these studies were conducted by Sunita Kanswal and Nora Katzenelson in my lab. Next, we wanted to understand if Taki was playing a role in other immune cells, and we focused on macrophage, and we wanted to describe the Taki phenotype in macrophage. And this project was conducted by Winde Alman in my lab. When we incubated macrophages with different toll-like receptor ligands, normally they mount an inflammatory response, as you can see here with the gray bars. All the TLR ligands can induce the production of TNF and IL-6. They also can produce, of course, the upregulation of CD40 and CD80, but we don't see this with Taki knockout mouse. So already we found that Taki knockout mice, macrophages don't respond to TLR ligands very well. And this seems to be because they express low levels of the receptors, toll-like receptors. Here you see a confocal microscopy of TLR4, and my D88 colocalization does not happen in Taki knockout mouse. So can they be, the macrophages of Taki knockout mice be M2 phenotype? So what does M2 phenotype mean? M2 phenotype in macrophages is the anti-inflammatory phenotype. M1, on the other hand, is the inflammatory phenotype. And LPS and interferon gamma, for example, is a very well described stimulus for driving macrophages to M1 phenotype, whereas IL-4 and IL-13 drives macrophages to M2 phenotype. And these two phenotypes, the polarized form of macrophages, in other words, are very important in different disease settings. For example, M1 macrophages are important in controlling intracellular bacteria, viruses. They are responsible for atherosclerosis and diabetes. On the other hand, M2 macrophages are anti-inflammatory. They play a role in responses to helminths, granuloma formation, cancer, tumor-associated macrophages, wound healing, and asthma is another example. So we wanted to characterize further the macrophages of Taki knockout mouse, and the one way to do is that measure the molecules that are associated with the M1 or M2 phenotype. Here you see examples of M2 molecules, IL-4 receptor alpha, which is expressed higher in Taki knockout mouse. CD206 also expressed higher in Taki knockout macrophages. And the gene expression here, you can see that IL-10, arginase, FIS1, IL-1 receptor antagonist are all expressed higher except YM1 in this experiment, although we do measure higher YM1 in some cases too. So this suggests that Taki may be transducing signals from BAFF and APRIL that drive the macrophages to M1 phenotype. And in the absence of Taki, this does not happen. So this is the working hypothesis. To test this, we incubated macrophages from wild type or Taki knockout macrophages from BAFF and APRIL, with BAFF and APRIL. And here you can see the result for the M2 phenotype molecules when interferon gamma and LPS is used as a control. This is the result for wild type mice. As the control suggests, interferon and LPS downregulates CCL22, which is the M2 phenotype. Just like this positive control, BAFF and APRIL also downregulates these M2 molecules. The results for the others are pretty much the same. And in the Taki knockout mouse, we don't see that because there's no ligand. BAFF, CCL22 doesn't change with BAFF and APRIL, although the positive control works. And what about the M1 markers? Here you see IL-6, the positive control. Here again, interferon gamma and LPS. And the Taki knockout mouse mounts much less IL-6 compared to wild type. Another example is MMP9. BAFF and APRIL both induce in wild type mouse, but not in Taki knockout mouse. So same with CD80, which then tells us that both BAFF and APRIL can suppress M2 markers and induce M1 markers. And in the absence of Taki, the levels of M2 markers increase. So how does this transduction occur? We know from the literature that interferon and LPS can engage several different signaling molecules. Here are some examples of them. This is a positive control, for example, JAK pathway, STAT1, STAT3, and ERK, and STAT6. What we found is that in the wild type, BAFF and APRIL both can induce phosphorylation of STAT1, which is an indication of the activation, and STAT3, and also ERK. In the Taki knockout mouse, STAT1 was diminished, indicating that it's involved, but not STAT3, and also ERK was diminished. So it seems like STAT1 and ERK are both involved in BAFF and APRIL-induced signaling. So we wanted to further interrogate this. We used an ERK inhibitor, U126. This is a chemical inhibitor of ERK, and this is a Western blot where you can see that BAFF and APRIL stimulated macrophages. Phosphorylation of this macrophages was inhibited with the inhibitor, so the inhibitor works. And then ERK is also inhibited. So this is an ERK inhibitor, but it inhibits phospho-STAT1 too. This is important. And as a result, these cells now secrete much less IL-6. MMP9 expression is decreased, which means that these two pathways are involved in the inflammatory response. We see the same thing with CD80 is a flow cytometry inhibitor, inhibits the upregulation of CD80. We also wanted to inhibit STAT1. There's no chemical inhibitor for STAT1, so we used an siRNA method to block the expression. And here you can see that the siRNA works. Our siRNA for STAT1 inhibits the expression of the production of phospho-STAT1. The control MAC and the control siRNA don't do this. And interestingly, ERK was not affected. Only STAT1 was affected. And here is the outcome. IL-6, the black bar here with the inhibitor for STAT1 is inhibited. MMP9, TNF-alpha, and INOS are all inhibited, indicating that indeed STAT1 is involved in BAFF and APRIL-induced activation. So what these experiments tell us is that ERK is upstream of the STAT1, and then both of them transduce signals to induce the expression of IL-6 and TNF-alpha, MMP9, and INOS. At the same time, arginase 1, FIS1, the M2 markers are downregulated. So these are the description of the phenotype of the Taki knockout macrophages. What does this mean? So we wanted to test the significance of these findings in 3 different models. First, I'll talk about the Leishmania infection model. Then I will talk about a high-fat diet-induced obesity and metabolic disease. And finally, if I have time, I'll talk about lupus disease. So Leishmania is a suitable model for testing macrophage phenotype because first, Leishmania infects macrophages, and in black 6 mouse, C57 black 6 mouse, what happens is that the IL-12 produced by dendritic cells drive the Th1 polarization of cells, which produce interferon gamma and kill the parasite inside macrophages through nitric oxide. And that's why black 6 mice are described as Leishmania resistant mice. On the other hand, BOP-C, which is a Leishmania susceptible mouse strain, dendritic cells produce IL-4. This IL-4 drives Th2 polarization, and IL-4 and IL-13 produced by Th2 cells then drive the M2 polarization of macrophages, which produces arginase and polyamines, help actually propagate the parasite. So that's why it's a good model. And this for this project, Wendy Allman and Rana Day from Office of Blood collaborated. They are experts in Leishmania infection. And we started by an in vitro experiment where we incubated macrophages from wild type and Taki knockout mouse with Leishmania promosticos. And here you can see that the wild type mouse under microscopy, the black dots here after 4 days of incubation, there are some parasites left. In the Taki knockout mouse, on the other hand, there are a lot of parasites inside the macrophages. So these macrophages, in the absence of Taki, are not able to control the infection. Here is the quantification of this microscopy data, percent infected cells, and also the parasites per 100 cells are much higher in Taki knockout macrophages. So they are not able to control. And why they are not able to control is because they seem to be defective in producing nitrate, which is an INOS indicator required for killing the parasite. So next, we wanted to see if BAFF and APRIL themselves can control the parasite propagation in wild-type mouse and in Taki knockout mouse macrophages. So after 6 hours of incubation of macrophages with the parasites, they have equal number of parasites, which suggests that the uptake of the parasite or infection of the macrophages by the parasite is not hampered in the absence of Taki. So both cells from both mouse strains have equal number. But after 2 days, we can see that the BAFF and APRIL incubated wild-type macrophages harbor much less parasite. Here's the positive control, interferon gamma and LPS, whereas the Taki knockout macrophages incubated with BAFF and APRIL still harbor a lot of parasites. And this is the quantification. At the top dashed line here, you can see Taki knockout macrophages, the levels don't change. When the wild type, they are decreasing in time with BAFF and APRIL. So this is in vitro. What about in vivo? So in vivo experimental model is the cutaneous infection model with Leishmania major. Here, intradermally in the ear of the mice, Leishmania metacyclic promosticos are injected in Taki knockout versus wild type. After we followed these mice for 12 weeks, and we monitored them for ear histology, cutaneous parasite count, cutaneous INOS production, and T helper response. So first thing we observed is that in the Taki knockout mouse, the disease was much more pronounced. You can see the lesion size measurements in knockout versus wild type mouse. As I was alluding in the beginning, wild type black 6 mouse strain can control the infection. And here, on the other hand, there is obvious macroscopic changes in Taki knockout mouse ears. And the histological examination supports this macroscopic observation. There's a thickness, increased thickness in the Taki knockout mouse, infiltration of the cells, more parasites harbored in the ear of the mice lacking Taki. And the iNOS production here in the brown staining is much more in wild type, suggesting that iNOS is responsible for the killing, controlling of the parasite in wild type, but not in Taki knockout. And we also collected the macrophages from the ear and characterized them for arginase 1, IL-1, RN, and YM1. And the Taki knockout macrophages express much higher, suggesting that they are M2 phenotype. So as I said, T helper polarization to 1 is very important in controlling the infection in black 6 mouse. So therefore, we measured the T helper phenotype of these cells from the draining lymph nodes. And you can see that as expected in the wild type mouse, at 2 weeks, there is high production of interferon gamma, which is the signature cytokine for Th1 response. And this percentage decreases in time as the infection is resolved. Taki knockout mouse, on the other hand, cannot generate interferon gamma. And this is the quantification of the interferon gamma positive CD4 cells. And Taki knockout mouse, of course, cannot mount. There was no difference in IL-4 production. So this means that there is not any, that the mouse don't have more IL-4 producing T cells, but they have less, they can't mount interferon gamma. So all these data suggested that macrophages may play a role, but it doesn't prove that macrophages are responsible, because our Taki knockout mouse is a global knockout mouse, so that all the cells are lacking Taki. So that means that any cell can be responsible. So to show or test if macrophages are important in this susceptibility to infection, we did an adaptive transfer experiment where we transferred the PBS or injected PBS or Taki knockout macrophages or wild type macrophages to Taki knockout mouse after infection with the ear, and wild type mouse served as control. And we followed these mice for 5 weeks. So you can see, immediately appreciate that the Taki knockout mouse that received only PBS still had pronounced pathology. Taki knockout mouse received Taki knockout macrophages also had pronounced pathology. On the other hand, Taki knockout mouse that received wild type macrophages now had much less pathology. And this is the control wild type mouse. Of course, as usual, has much less pathology, and this is the quantification showing that when we transfer wild type macrophages into Taki knockout mouse, we can control the infection, and this is the parasite number harvested from the ears. The third column here, Taki knockout macrophages, wild type macrophages injected into Taki knockout mouse now harbor much less parasites, suggesting that macrophages by themselves are able to control the infection. So what happened to the T cell response after the transfer of the macrophages? Here you can see on the interferon gamma plot, the mice that received wild type macrophages now were able to produce interferon gamma just like the wild type mouse. Suggesting that this Th1 polarization can occur after the transfer of macrophages. So this is interesting because normally it was thought that the Th1 response was driving M1 phenotype of the macrophages, but our transfer experiments suggest that the opposite is also true. When we transfer macrophages, which are wild type, which are prone to M1, they can then drive Th1 response also. So in this part of the talk, the study, what we have learned is that the Taki knockout macrophages are M2 skewed. They engage MAP kinase and STAT1 pathway. They can control Leishmania infection. The control of Leishmania infection is limited in Taki knockout macrophages. And also the mouse cannot resist the infection. There is a deficiency in generating Th1 response. But when we transfer wild-type macrophages, Taki-positive macrophages, we can control the infection and they mount Th1 response. You can get more information from this study in the publication listed here. In the next part of my talk, I'm going to focus on high-fat diet-induced obesity model. And this is an interesting model because obesity due to high-fat intake was initially thought to be, of course, it leads to diabetes, type 2 diabetes, solely endocrine phenomenon. But about 3 decades ago, a paper published by Gökhan and his mentor Bruce Spiegelman showed that actually immune system is also involved. They showed that TNF alpha produced in adipose tissue is responsible for the resistance of the adipocytes to insulin. So after this publication, there was an influx of study by the immunologist, which led to the characterization of adipose tissue immune system involvement. So in short, in the lean adipose tissue, which is insulin sensitivity is high, there is the adipocytes here depicted with the as yellow cells surrounded by M2 macrophages in green here. There are also eosinophils which are responsible for IL-4 and IL-13. In contrast, after high-fat diet intake, obese adipose tissue, the adipocytes enlarge and then start to produce chemoattractants, which then chemoattract many different cells, but the M1 macrophages are very important in the disruption of the homeostasis in the adipose tissue, depicted with the red cells here. And they form this crown-like typical structure. There's a lot of inflammatory cytokine secretion in this tissue. So that's why it's a good model. And the model works like this. But first, I would like to introduce Lunha Liu here, who has conducted this study heroically, because my lab was not a high-fat diet model studying lab. So in this model, then mice are fed 14 weeks with a high-fat diet or normal chow diet, NCD. And then we wait for the mice to gain weight. And then first thing we observed by comparing these two strains is that, as expected, the wild-type mouse here in red gained weight compared to normal chow diet in black here. In contrast, even though Taki knockout mouse also gained weight, the weight gain was much, much less than the wild-type mouse. And when we analyze the subcutaneous and visceral adipose tissue, while the wild-type mouse fed on high-fat diet had enlarged adipose tissue, Taki knockout also had enlarged adipose tissue, but it was much smaller than the wild-type mouse. So typically, the way to study the effect of high-fat diet on glucose metabolism, insulin metabolism, is that you perform glucose tolerance test and insulin tolerance test. In these tests, mice are injected with glucose or insulin, and the blood glucose levels are measured. As expected in red here, the wild-type mice that received high-fat diet now had trouble clearing glucose. I hope you can appreciate this obvious delay. And in the Taki knockout mouse, blue here, on the other hand, glucose was cleared much faster. Similarly, after injection of insulin, glucose clearance was delayed, decreased in wild-type mice, but in the Taki knockout mice, this clearance was as good as the normal chow diet fed mice. And we wanted to see if macrophages played any role in this. First, we harvested adipose tissue macrophages. Here in the left side of the bar graph, you can see that the Taki knockout mice, black, compared to gray, wild type, after high-fat diet feeding, they had much less macrophage accumulation. The M2-like macrophages were more in Taki knockout mice fed with high-fat diet. On the other hand, the M1 macrophages were higher in the wild-type mouse fed with high-fat diet. CD206 is a marker of M2 phenotype, and it was higher in high-fat diet fed Taki knockout mouse macrophages compared to wild-type mouse fed with high-fat diet. Here are genes that are representative of the M1 and M2 markers. As expected, wild-type mouse had high TNF, and compared to Taki knockout mouse. On the other hand, IL-10, for example, this is the same for all the genes, so I'm just focusing on one, was higher in Taki knockout mouse macrophages and lower in wild-type after high-fat diet, suggesting that the macrophages phenotype in the adipose tissue were M2 in Taki knockout mouse after high-fat diet feeding. And so again, these experiments tell us about the global effect of the high-fat diet feeding, but it doesn't tell anything about if macrophages were responsible, because, for example, B cells, B2 cells, are important in driving inflammation in the adipose tissue. T cells can be responsible also. So to address the role of macrophages, then we again performed adaptive transfer experiments. In these experiments, as depicted here, high-fat diet fed mice were transferred with macrophages isolated from the peritoneum of either wild-type or Taki knockout mice, which are also high-fat diet fed, and then intravenously, and then monitor these mice. You can see the groups here. So we have the normal chow diet fed wild-type mouse, which is the control, high-fat diet fed wild-type mouse injected with PBS, or mice injected with macrophages from high-fat diet fed wild-type mice. And then, of course, this is our test group, Taki knockout high-fat diet macrophage, also injected. First, we analyzed the serum levels of insulin. In mice that received high-fat diet PBS, there was high insulin. This is a Taki knockout macrophage mice, so that is expected. And when we injected more wild-type macrophages, there was even further increase in insulin levels. But on the other hand, when these mice received Taki deficient macrophages, the insulin levels went down. We saw the similar thing with the IL-6 and TNF. As you can see, IL-6 levels are much higher in mice that received Taki deficient macrophages, blue here, whereas transfer of Taki intact macrophages then further increased IL-6 levels. Similarly, TNF levels were also low in mice that received Taki deficient macrophages. So what happened to the GTT and ITT, the tolerance tests? So here you can see that the blue is the control here, wild type high-fat diet. As shown previously, they have difficulty clearing glucose. And when these mice received more macrophages from wild type, there was a slight increase, but this was not statistically significant. But more importantly, when the Taki deficient macrophages were given, there was a decrease in glucose clearance. So increasing glucose clearance, so decreasing glucose levels. We saw the same thing with insulin tolerance test, glucose levels were cleared much faster after the transfer of Taki deficient macrophages, suggesting that just the macrophage transfer is sufficient to alter and correct the insulin resistance. So talking about insulin resistance, so how can the Taki deficient mouse maybe providing help or to increase the insulin sensitivity? To test this, we harvested the media from Taki deficient macrophage cultures or wild-type macrophage cultures and incubated them with adipocytes and then stimulated with insulin. And typically, insulin induces the phosphorylation of three molecules, IRS1, AKT, and ERK. And you can see the control in the Western blot. The phosphorylation is depicted. And when we wild-type mouse macrophages media is added, this phosphorylation is decreased slightly. In contrast, when the media from Taki knockout macrophages is added and cells are stimulated with insulin, we now see an enhanced phosphorylation of IRS1, AKT, and ERK. And this is the quantification of the Western blot. And this suggests that the whatever is secreted by Taki knockout macrophages, it renders them more sensitive to insulin, which helps explain why they are protected from high-fat diet with obesity and metabolic disease. And so to understand which molecule may be responsible for eliciting sensitivity to adipocytes, we measured the TNF-alpha and IL-6 levels. You can see that the high-fat diet fed wild-type mouse have high TNF, and Taki knockout mouse have slightly increased, but still lower than the high-fat diet fed wild-type mouse. IL-6 has the same phenotype. In contrast, the anti-inflammatory cytokine IL-10 is significantly suppressed in wild-type mouse after high-fat diet, and in the Taki knockout mouse there is also a decrease, but this is still much higher than the wild-type mice fed with high-fat diet. So IL-10 can be a candidate as a cytokine that is helping sensitize adipocytes to insulin. And to test this, we used blocking antibodies to IL-10 and performed the same Western blot, measuring IRS1, AKT, and ERK phosphorylation. Here's the control. As expected, insulin induces the phosphorylation of these molecules. So if we just add the Taki deficient medium, there's a slight increase in the phosphorylation of this molecules. And this, when we use the control IgG, this increase in the phosphorylation of these molecules doesn't change. But when we add anti-IL-10, we almost completely block the phosphorylation of these molecules, suggesting that IL-10 may be responsible for the sensitization to insulin. So this is our working hypothesis model. In obese mice, these are adipose tissue cellularity, and one macrophages dominate. They block insulin signaling, most likely with TNF, as Gökhan has shown. And then when we transfer Taki knockout macrophages, they home to adipose tissue. I didn't show the data, but we can show that the macrophages in the adipose tissue are coming from Taki knockout mouse after the transfer. I also didn't show data, but the eosinophils also accumulate here. This is important because eosinophils provide IL-4, and these cells then, by producing IL-10, sensitize adipocytes to insulin. And here, again, I didn't show data, but we did detect the polarization of host macrophages to M2 also, and we believe it's through the IL-4 coming from the eosinophils that influx into the adipose tissue. So this is our working hypothesis for the for the obesity model. And in the last part, I'm going to shortly talk about a more recent paper on lupus disease and Taki. So lupus disease briefly is an autoimmune disease, involves autoreactive B cells and T cells. Mostly women suffer from childbearing age, suffer from this disease. And typically autoantibody produced forms immune complexes, and they deposit in the skin and kidneys and cause damage. And you can see here the typical butterfly rash, skin lesion, and the kidney deposition of the immune complexes causes a very important outcome. So potential targets for SLE treatment has always been so far blocking the B cells, because B cell produced antibodies are responsible for the disease. For example, in 2011, a monoclonal antibody against BAFF was licensed and currently used in SLE treatment, which basically wipes out the B cells, autoreactive B cells, for a while. And previously, another B cell targeting drug, rituximab, was approved in '97, again targeting B cells. So there are several different mouse models to study lupus. We used MRL-LPR mouse. In this strain, the LPR version of the MRL mouse has a mutation in the FAS, which is apoptosis signaling molecules, so the B cells don't die, and they continue to, the autoreactive B cells continue to produce antibodies. So what we did is that we backcrossed this MRL-LPR mouse to our Taki knockout mouse to generate a lupus mouse deficient in Taki. And here is the plus, minus, and minus versions of these mice. You can see that the plasmablasts and plasma cells in the plus produce Taki, plus minus heterozygous produce less, and there's no Taki expression in the minus version. So the first thing we observed is that these mice typically develop disease and they die prematurely because of the kidney failure. And first thing we observed is that the Taki deficient lupus mouse survive more. And I'm not showing the data, but they had less lymphadenopathy development. So this is the obvious first outcome. It seems like the Taki deficiency leads to survival of the mice. And typically, the development of the disease is monitored by measuring serum anti-double-stranded DNA and RMP antibodies. You can see that the development of these antibodies are delayed in Taki deficient mouse. After 12 weeks, both the strains have comparable levels of the antibody, but there's a significant delay. And this may explain why there is a delay in also the death of the mice. And you can see other evidences of this decreased antibody immunofluorescence staining also. So what happens to the proteinuria? The kidney functions, again, there's a delay in the proteinuria score. After 16 weeks, they seem to be, both the strains catch up, but there's a significant delay. You can see the diminished infiltration of the cells in the glomerulus, and this is quantified. Here's a histology of histopathology of the plus and minus mice. And also there is less deposition of immune complexes as measured with IgG and complement 3 here in the minus mice, further supporting the evidence for the why these mice are surviving longer. And we harvested the macrophages from the kidney, so we know that the antibody response is less, but what about the macrophages? And first of all, first thing we saw is that there's less macrophages in minus mouse kidneys. And these macrophages manifest an M2 phenotype as measured with IL-4 receptor alpha, CD206. And then they had less M1 markers. And here are the other markers, the genes that are representative for M1, TNF-alpha, INOS, IL-6. They're all less in minus mouse, and M2 markers are higher. So suggesting that in addition to low levels of antibodies produced in the absence of Taki, the macrophages are also M2 phenotype, and there are less in the kidney, so that may be helping in preserving or extending life. So again, we did an adaptive transfer experiment. In this case, we transferred Taki knockout macrophages into wild type MRI-LPR mouse, lupus mouse. Here are the groups: control, nothing transferred, PBS transferred, macrophages from plus mouse transferred, or macrophages from minus mouse transferred. So when we, the timing of the transfer is indicated with the arrow here. After the transfer, immediately we see an improvement in proteinuria score, but this doesn't last long. So when we transfer again macrophages, again we see a decrease. So temporarily, transiently, macrophages alleviate the kidney proteinuria. And this is the histology staining. You can see in the plus transferred mice or minus transferred mice, there is a significant difference in the pathology. And here's the score for this. So these experiments in the lupus mouse then again showed us that there's a delay in autoreactive antibody production in the absence of Taki, and we believe this is a T-cell independent antibody production delay, because the data that I haven't shown, both the mouse strains have mounted comparable levels of T follicular helper cells and germinal center response, suggesting that the T-dependent response was not affected. And then we also showed that ablated M1 macrophage influx into the kidney may be responsible for the extended life. So what is the implication overall of the work we have done on Taki, and especially in the role of Taki in macrophage phenotype, because this was not described previously? First, as I said in the beginning, newborn age, there's very low levels of Taki expression on B cells. I can say that macrophages also express very low levels of Taki. So this may mean that the phenotype of the newborn age of the macrophages may be due to low Taki expression, and this needs to be explored, and we are currently working on that. I mentioned that combined variable immune deficiency patients with Taki mutations, exist, and their B cells are well characterized, but their macrophages are not characterized, so their macrophages may be M2 phenotype. Indeed, some patients, especially young patients, appear to be prone to asthma, and this may be due to the M2 phenotype of the macrophages. Finally, the data from the high-fat diet experiments suggest that Taki inhibitors may help alleviate obesity-induced metabolic disease. So Taki can be a target to treat obesity-induced disease. And finally, I would like to say that this is the Taki. I'm sorry, there was maybe a disconnection, but I hope I'm still on. It seems like Taki assumes a new role every day, and this role is changing every day, and it seems like the final territory for Taki is in the culinary business, and we can see that in Turkish restaurants, they have a role, and even Italian restaurants, Taki seems to be playing a role. And with that, I would like to thank the people in my lab who have contributed to the work I described here. And we had very productive collaboration with Hira Nakashi's lab at FDA, and also we collaborated with Gökhan for the, I'm sorry, so are we still on?
Sununuzu göremiyoruz Mustafa Hocam. Sesinizi duyuyoruz ama.
Hala göremiyor musunuz?
Evet,
Şu an
sanırım o bilgisayarda bir sorun oldu. Sunumunuzu şu anda göremiyoruz. Sadece sesinizi duyabiliyoruz.
Kötü olmuş. O zaman evet şeyden kopmuşum ben. Zoom'dan burada. O zaman tekrar Zoom'u belki açabilirim. Onun için linke tıklamam gerekecek. Ama yapamıyorum. Neyse. Sonuçta umarım bir saniye belki tekrar bir umut oldu. VPN connection problem diyor da evden bağlanınca böyle şeyler olabiliyor maalesef. Biz bir de FDA sistemine dahiliz VPN'le. Oradan da kontrol var. Ama herhalde bağlanıyorum tekrar. No, computer sesi kapatalım. Mute. Görünüyor mu şimdi acaba?
Evet, şu anda görebiliyoruz.
Güzel. En önemli kısmı atladık. Sonra şurada implications diyordum. Herhalde burada koptu.
Evet.
Newborn macrophage phenotype determination is important. Combined variable immune deficiency patients important. And then Taki may be a target for treating obesity-induced metabolic disease. And the new role for Taki, I was really hoping that I could show this slide, and we can see that the Taki now plays role in Turkish culinary. Taki's Beiti, Tachy of course is pronounced, and also in Italian culinary. So the role of Taki is expanding to many different directions. And here are the people who did this work in my lab over the years. And here on Akashi at FDA, Gökhan Tamışlıgil at Harvard helped us with the metabolic cage experiments, and I am in debt to Richard Bram, who gave me the Taki knockout mouse, and also thanks to the core facility. Okay, thank you.
Çok teşekkür ederiz. Kameranız da galiba kapalı Mustafa Hocam. Tamam, şimdi görüntünüz geliyor. Bu güzel konuşmanız için çok teşekkür ediyorum ben öncelikle. Soruları Türkçe olarak almamız uygun mu sizin için? Nasıl devam edelim? Nasıl isterseniz?
Olur. Tabii olur.
Bir tane sorumuz var aslında izleyicilerimizden. Leishmania konusunu anlatırken şey demiş, are the same events used in cancer treatment? Yani kanserde benzer bir mekanizma oluyor mu diye merak etmiş diye anlıyorum ben. Taki knockout'larda, kanser background'ında veya kanser modelinde.
Kanserde şu ana kadar en önemli Taki ya da buff sistemi diyelim şeyi.
Galiba bağlantımız koptu.
Bilmiyor mu?
Şimdi sesiniz geldi. Tekrar edebilirseniz bir bağlantınız durdu.
It says your connection is unstable. Duyuluyor mu şimdi?
Şu anda duyabiliyoruz.
Tamam.
Ama en önemli şey dediğiniz orada kesildi.
Tamam. En önemli şey, şimdi normal fizyolojide BCMA 3 reseptörü var demiştik BAFF and APRIL sisteminin. BCMA sadece plazma hücrelerinde bulunuyor diye ve çok da fazla çalışılmadı. Ama multiple myeloma'da BCMA çok önemli. Artıyor ve sorumlu myeloma hücrelerinin gelişmesinde. Ve hatta şimdi klinik çalışmalar BCMA'yı target eden monoklonal antibody'lerle önemli bir şeye geldi, faza geldi. Ama Taki'nin direkt olarak kanserle ilişkili çalışmaları çok az. Yani yok diyebilirim.
Anladım. Benim de birkaç sorum var aslında. Leishmania enfeksiyonlarında aynı zamanda TH17, TH17 hücrelerinin de etkisi olduğu biliniyor. Ben onu siz anlatırken o geldi aklıma. Acaba TH17'lerin durumu nedir Taki knockout'larda?
Tipik bir şey yok, deficiency. Ama Leishmania'da da baktık biz, orada da fazla bir değişiklik yok. Esas değişiklik T-helper 1 and TH2. Yani
peki B hücrelerinde ve antikor üretimi nasıl, ne durumda?
baktık. Antikor üretiminde de bir değişiklik yok ama izotype'lar farklı. Dolayısıyla yani tek izotype'lar var. Tamamen.
Bir de ben
Pardon.
regülatör vericilerle çalışıyorum. O yüzden onu da merak ettim. Hiç interleukin üreten bireklere baktınız mı?
Var tabii. O yani başkaları gösterdi onu. Taki deficient B hücreler, B-reg hücreler genellikle.
Okay.
Zaten
O zaman bir sonraki sorum tabii bunu takip ediyor. O zaman diğer modellerde hani makrofajları gösterdiniz. En son özellikle bu SLE modelin hastalığında. Orada tam etkiyi makrofajlar gösterilmiyor. Sadece bir delay oluyor, gecikme oluyor. Acaba orada bireklerin de bir rolü olabilir mi?
olabilir. Olabilir. Bakmadık ona. Ama olabilir. Olabilir. Şeye baktık mesela. T-reg'lere baktık. Çünkü o T-reg'ler de orada bir fark yoktu. Olabilir. Tabii yani daha fazla incelenmesi lazım o modelin. Ama SLE zaten çok kompleks bir disease. Yani bir şekilde geçici olarak makrofajlar yardım sağlasa da başka faktörler diyelim tamamen o geri döndürüyorlar. Uzun süre yapamıyor. Ama dediğim gibi belimumab mesela buff monoclonal antibody o da geçici çalışıyor aslında yani hastalarda geçici bir süre rahatlık veriyor ama tekrar B hücreler tedavi kesildikten sonra çünkü uzun süre yapamıyorsunuz tabii tedaviyi çünkü o zaman immunodeficiency olacak. Öyle bir var, engel var maalesef. SLE'de. SLE çok challenging. Bir şey söyleyebilirim. Mesela B-reglerle söylediniz. Şu anda çalıştığımız dedim ki yeni doğanlarda Taki az ve yeni doğanlarda enteresandır. B hücreleri de çoğunlukla B-regulatory, IL-10 producing. Şimdi onu çalışıyoruz. Hangi mekanizmayla olduğunu.
Taki az olması lazım. Peki mesela Taki ile BAF reseptörünün bir arada knockout'unu hiç denediniz mi? Yani o iki yani reseptör BAF'ın veya APRIL'ın bağlanabildiği şeyler olmazsa nasıl bir etki olur?
Şöyle olur. Şimdi o BAF reseptör ilk önce ekspres oluyor. Taki yok. Şeyler mature olurken, olgunlaşırken B hücreleri ilk BAF reseptör ekspres oluyor. O sırada Taki yok hücrelerde. Dolayısıyla BAF'ı zaten knockout ettiğiniz zaman Taki olan hiçbir hücreye geçememiş oluyorsunuz.
Anladım.
Development'ta yani overkill oluyor.
Belki conditional olarak induce edilebilir sonrasında. Yani Taki conditional knockout yapılarak Taki önce ondan sonrasında BAF sonrasında şeklinde.
O zaman olabilir. O zaman da yine ölecekler ama hücreler. Yani
Bir noktada ölecekler. Doğru.
Olmadığı zaman hücreler hemen ölüyorlar. O survival signal.
Çok enteresan. Özellikle yani obesity'deki çok makrofajlar ve innate hücrelerin adipose dokuda olduklarını daha önce Gökhan Hoca'dan da dinlemiştim birkaç sefer. Orada ama çok fazla adaptif edinsel hücreler çok yüksek miktarda değil herhalde değil mi? Genelde hep innate hücrelerin adipose dokudaki daha ön planda. Doğru mu düşünüyorum yoksa?
Şimdi çalışmasına göre değişiyor. Yani öyle çok güzel çalışmalar var. Direkt olarak özellikle CD8, effector CD8 T hücrelerin inflamasyona katkıda bulunduğu gösterildi. Yani o knockout mouse'larla falan bayağı güzel şeyler var, bulgular var onda. Biz de gördük CD8 hücreler artıyor wild type'da hakikaten ama Taki knockout'ta azalmadılar. Yani bizim modelimizde onların bir rolü yok. Çünkü hala vardılar ve bizim modelimizde direkt olarak yani bence adaptive transfer deneyleri hakikaten bayağı inandırıcı. Çünkü B hücreleri de transfer ettik. Mesela yani onlar da rol oynuyor mu acaba diye B2 hücrelerini. Onların hiçbir etkisi olmadı. Aynı şekilde devam etti hastalık. Sadece Taki eksik olmayan makrofajlarda görebiliyoruz iyileşmeyi.
Bir de aklıma geldi. Adaptif transfer deneyleri yaptınız ama acaba hiç kemik iliği nakli denediniz mi? Yani sadece onların olmadığı bir background olması acaba önleyici olur mu? Hani baskılaması değil de oluşmamasını sağlar tarzda bir düzeniniz oldu mu?
Olmadı ama daha iyisi var. Şöyle conditional knockout yaptık şimdi. Yani sadece makrofajlarda Taki yok. Sadece B hücrelerde Taki yok.
Evet, en güzeli. Evet.
Hangisi? O var. Şimdi ona çalışıyoruz. Evet.
Anladım. Çok çok çok teşekkür ederim. Çok gerçekten güzel ve benim için de çok ilgimi duyduğum bir konu olduğu için çok zevkle dinledim. Şimdi baktığımda başka bir soru şu anda göremiyorum. Ben size çok teşekkür ediyorum gerçekten zamanınızı ayırdığınız için. Ve aynı zamanda Türk İmmünoloji Derneği olarak da tekrar teşekkür etmek istiyorum davetimizi kabul ettiğiniz için. Size orada iyi günler dilemek istiyorum. Çok
Evet,
teşekkürler.
iyi akşamlar. Esas ben teşekkür ederim. Çok güzel bir şey oldu benim için, fırsat oldu. Umarım konuşmam şey yapmıştır, bir stimulus olmuştur. Dediğim gibi özellikle Combined Variable Immune Deficiency Türkiye'de çok hasta var
Evet.
Taki deficient. Onlarda makrofajlar, fenotipleri çalışılmadı. Yani o konuda belki kolaborasyon yapmak isteyen olursa yani umarım başka şeylere de yol açar konuşma.
Tabii mutlaka. Ben ona inanıyorum. Çok teşekkürler. Çok sağ olun Mustafa
Ben
Hocam.
teşekkür ediyorum. İyi akşamlar. Tekrar sağ olun.
Sağ olun size de. Hoşça kalın.